coffee and get here on time. Yeah, it's fine to start now. No, I think it's okay. So we are now gonna shift gears from the very nice presentation from Professor Rollins into exploring highly heterogeneous conformational ensembles through experiment and simulation. So the very first speaker will be Professor Ben Schuler, and then we are going to have a second short talk from selected posters. So Professor Ben Schuler is a professor of molecular biophysics at University of Zurich, and he has obtained his education and training in biochemistry and biophysics, working in several institutions in the UK, Germany, and the US before settling in Switzerland. He has been working with single molecule techniques applied to the dynamics of disordered molecular systems. And today he's going to be talking to us about probing the rapid chain dynamics of disordered proteins and nucleic acids. So Professor Schuler.
Yeah, thank you very much. And thank you very much for this wonderful invitation to Brazil. We already had a wonderful meeting last week, and it's been fascinating to be here. And the hospitality makes me feel like a member of this wonderful Brazilian family of science. And when I came here, I realized that there's even a direct flight from Zurich to Sao Paulo, so I clearly should come more often. It's been wonderful. And so, yeah, I will focus on the rapid dynamics of disordered proteins and nucleic acids, and I would like to focus on how we can measure these properties experimentally using single molecule spectroscopy. But sort of to put this into a bit of a broader context, I would like to summarize the types of different kinds of dynamics that we can measure with single molecule experiments before I go into more depth about the rapid dynamics. And so if we illustrate this with an example of coupled folding and binding, where we have here, is my pointer work, an intrinsically disordered protein, immobilized here on a surface, and when it binds its binding partner, it folds into a well-defined complex. And we can investigate this with Fred experiments by attaching a donor chromophore to one of the two proteins and an acceptor chromophore to the other. And then when they come together in the complex, then we get energy transfer from the donor to the acceptor. And so if we read this out experimentally, we can get time traces like these, where we now look at the number of photons per time as a function of time that we can measure. And so in this example, you now see this anti-correlated signal between the acceptor emission in red and the donor emission in green. And so that means that every jump that we see here corresponds to a binding or a dissociation event. And one way of analyzing this now is to look at the distribution of times that the system spends either in the bound state, that would be our tau f, or folded in this case in the bound state, and the times in the unbound state or unfolded state. And you could, with my students in a basic lecture, I actually asked them to do this by hand, to just take a ruler and measure these times, and then from the distribution of these dwell times in the states, you get the kinetic information about the system. Right? You can also use your favorite algorithm, for instance here, the Viterbi algorithm, to get such a state trajectory, and then from this automatically extract the dwell time distributions right? And get these decays, and then from the rate constants, from the decay constants of these curves, you get your rate coefficients, right? Now, once you've settled on a model, the way we do this in the lab is using Markov state models, and then you maximize the likelihood to get the correct parameters. So there's a whole field of people that focus on these kinds of experiments that describe sort of the classical chemical kinetics of such a process. Now, one of the simplifications in this approach, of course, is, for instance, that it assumes that these jumps between states are instantaneous, which, of course, they are not. Right? And so a slightly different way of looking at this problem would be to treat this in terms of diffusion on a potential of mean force. And with Peter and Jose, of course, we have some of the pioneers here who showed us that even for a reaction as complex as protein folding, thinking about this process in terms of diffusion on a relatively simple potential of mean force can still be a very useful and meaningful approach. And so if we have this potential of mean force, right, then our molecule would sort of go back and forth between here, the unbound or unfolded state, and the bound or folded state here. And then every off and on, it would jump across the barrier. And if you monitor this as a function of time, right, you could have here the dynamics in the folded state, then the jump across the barrier, and then you're in the unfolded state. And of course, the jump across the barrier takes a finite time, and we call this the transition path times of this process. And this is also something that we can get from single molecule experiments to some extent. And so this is an approach that was pioneered by Bill Eaton and Hoi-Sung Chung at the NIH. And so their idea was, let's now focus in on one of these transitions. Instead of binning the photons in a relatively coarse fashion as it is depicted here, you now look at the individual photon arrivals, because with the experiments we do, we monitor every photon individually with picosecond time resolution. And so in principle, we have very good time resolution, but what limits us is the number of photons that we can collect during such a transition. But if we plot now here in green the donor photons and in red the acceptor photons, you can see how we go from a high photon rate in the acceptor to a high photon rate in the donor channel. And so in principle, the information about the time it takes to jump across the barrier is encoded in these data. The question is how to get this information out. And so here, the elegant approach that Bill Eaton and Hoi-Sung Chung came up with is to say, okay, let's compare two simple models. Let's compare the model where we have an instantaneous jump from the folded to the unfolded state to a model where we introduce a little virtual intermediate here that essentially corresponds to something like the transition state. And then we vary the time that the system remains in this intermediate state, and then we calculate the difference in the likelihood between these two scenarios based on these data. And if we then look at this delta log likelihood as a function of the duration of this intermediate here, then if we have enough time resolution, we get a peak here in this delta log likelihood function, and the time scale of the peak tells us the approximate duration of this transition path, which for protein folding, these transition path times tend to be in the range of microseconds. That's something that we've also been able to do for such a coupled folding and binding reaction. And the challenge is to get enough photons to do this, but there's recent efforts using nanophotonics to enhance the emission to a level that we can get photons in a range of 10 megahertz count rates. And so there is hope that we will be able to actually resolve these transition paths, which are, of course, some of the most interesting pieces of the whole trajectory, because that's where the actual action happens. Okay? So now we have kinetics of interconversion. We have an idea about the time it takes to cross the barrier. But there's, of course, more dynamic information that we would like to know. So, for instance, the dynamics within the folded state and within the unfolded state. Now, it turns out to get the dynamics in the folded state, single molecule experiments are not, the single molecule fluorescence experiments are not that well suited, because under these conditions, we will be dominated by the flexibility of the fluorophores, and so it's very difficult to get information about the dynamics within a folded protein unless there are large conformational changes that we can probe. But what we can do very well is to probe the dynamics in the unfolded, or in this case, the unbound state, because here we have large distance fluctuations of such an unfolded or disordered system. And so we can measure this by now looking at the fluctuations in fluorescence emission from our donor chromophore and our acceptor chromophore, right? Because as they come closer together or move apart, we expect that the emission from the donor and the acceptor will fluctuate on the time scale on which the distance fluctuates, right? And so we can measure these dynamics in terms of these kinds of correlation functions, right? So we correlate the signal that we get from the donor and the acceptor. So here in this example, in red, you would have the autocorrelation of the acceptor signal. In green, the autocorrelation of the donor signal. And in blue, you have the cross-correlation between donor and acceptor, right? And so the autocorrelations have a positive correlation. The cross-correlation has a negative correlation, right? Because as they get closer, for instance, right, the acceptor emission will go up and donor emission will go down. And if they move apart, the opposite will happen. So you expect them to be anti-correlated, and this is why we see this characteristic pattern in the correlation functions. Now there's also a dip here in the correlation function at very short times, and this is the signature of a single quantum system, right? So if you have an individual molecule or an individual fluorophore, you can never get two photons at the same time. And so this means that this correlation function, which you can think of as a conditional probability of seeing two photons, has to drop here at short times. And this photon anti-bunching, as we call it, we'll come back to that a little later today. So this sort of summarizes some of the key aspects of protein dynamics that we can measure using single molecule fluorescence experiments. And now I would like to focus on examples where we've been using this approach up here, this nanosecond fluorescence correlation spectroscopy, as we call it, to investigate different disordered systems. But let me, before I go into some of the examples, mention one other aspect, because if we now have these correlation functions measured, strictly speaking, what we've measured here is, of course, not the actual distance correlation function, right? What we want is we want information about the distance. What we've measured here is a fluorescence intensity correlation function. So we still need to go from fluorescence intensity to distance, and this is not completely trivial because we have this nonlinear dependence of our signal, the transfer efficiency, which is just essentially just the probability of energy transfer from the donor to the acceptor, has this R to the 6 dependence on distance. And so we also need information about the average distance between our molecules. And so for this, we take advantage of measuring this transfer efficiency. And so for instance, here, if we have a system that can be in an unfolded or disordered state and in a folded state, we can now focus on this disordered part, or the unfolded part, and get the transfer efficiency, right? And so from the transfer efficiency, we have information about the distance. But since we're dealing with a disordered system, we have a distribution of distances, not a single distance, which complicates this analysis. And it turns out that we can get information about the distance distribution by combining this transfer efficiency that we can measure on the one hand, just from the number of photons that we can count, and the fluorescence lifetime, so the time that the molecules, our fluorophores, remain in the excited state. And if we combine this lifetime information with the transfer efficiency, then it turns out that if we had a single static distance, it would lie on the straight diagonal line here. And the fact that our signal for our disordered protein is away from the diagonal, this is a signature of a distance distribution that is sampled in the system. And so in practice, what we often do is we use simple polymer models, which work pretty well for disordered proteins, and we use those to describe the distance distribution within our unfolded or disordered molecules. Now, once we have the distance distribution, we can simply convert that into a free energy profile again, right? And so now we treat the system in terms of diffusion in such a potential of mean force, right? So we're giving up some information about the polymer properties of the system, but it turns out to be a pretty good approximation. And so now we have a great approach that was developed by Irina Gopic and Attila Szabo that allows us to calculate these correlation functions that we've measured experimentally based on the shape of our potential of mean force and something like an effective diffusion coefficient or relaxation time within this potential. And so essentially what we're doing, we're combining the dynamic information from our correlation experiment with the distance information from this single molecule Fred measurement. And this allows us to quantify both the equilibrium distributions of our system and their reconfiguration dynamics. So this is how we're combining the two pieces of measurements, or two types of measurements for characterizing these rapid dynamics of disordered systems. And so the first example I want to mention here, and this is a system that I've already talked about on Friday for those of you who were around at the meeting last week, we have these two highly charged disordered proteins. One of them is histone H1. This is not one of the core histones around which the DNA is wrapped, but it's the histone that sits on top of the nucleosome and sort of stabilizes the nucleosome structure, and on a larger scale, it leads to chromatin condensation. And if you look at the sequence here, you see that it's full of lysine residues. It has a little folded domain, but large parts of the protein are disordered and has a net charge of plus 53, highly positively charged disordered protein. And this is its chaperone in the nucleus. And so this highly negatively charged disordered protein, what it actually does in the cell is it helps to displace histone H1 from the nucleosome. The affinity of H1 to the nucleosome is extremely high in the femtomolar range. And so with this guy, dissociation is sufficiently rapid to be biologically useful. And this is an example of molecular stripping, if you like, Peter mentioned, or competitive substitution, as it's also called in the context of these disordered systems. And so these two proteins have been known to interact for a very long time, but it was unclear when we started with this work what the complex between the two would look like. And so in a combination of our single molecule experiments with circular dichroism spectroscopy, NMR with beta-crocholint, and simulations from Robert Best, this is the picture that emerged from our results, that when these two proteins bind to each other, which they do with picomolar to nanomolar affinity, they remain disordered. You still have here the little folded domain of H1, but you can actually chop that off, and they still bind to each other with picomolar affinity. And now we can use our nanosecond FCS experiments to probe the dynamics within this disordered complex, and this would be the example of such a measurement. So we take this population of the molecules in the bound state, and we use all of the photons in this population to calculate these correlation functions. And then if we convert that to a distance correlation time, we end up with a time scale of about 100 nanoseconds. So that means, so the long range motions in this disordered bound state are on the order of 100 nanoseconds, very rapid even in this bound state between the two molecules. Now, as I already mentioned on Friday, for those of you who were there, an aspect that we've been investigating more intensely over the last few years is a process that these two proteins undergo if you go to high concentrations. Right? If you go to sufficiently high concentrations, they actually phase separate and form droplets that are similar in spirit to many of these biomolecular condensates that I'm sure you've heard about over the last few years. And for highly charged polymers in this context, you speak of complex coacervation. And if you do this for H1 and prothymosine alpha, you get these droplets within which we have very high protein concentrations in the range of about 200 milligrams per milliliter. And the viscosity of these droplets is very high, on the order of 300 millipascal seconds. So for those of you who are not so familiar with viscosity, that's about 300 times more viscous than water. And so this was one of the key challenges that we faced over the last few years, to try and get information about the dynamics of these disordered molecules inside these droplets. And just very briefly, I would like to summarize the key result. And this is work done by Nicola Galvanetto, a fabulous postdoc in the lab. The idea is to phase separate the system. So you mix H1 and prothymosine alpha, unlabeled protein, and then you add a little bit of labeled protein that allows you to do FRED experiments, single molecule FRED experiments on these molecules inside the condensates. And then you can either measure outside the condensates, and then you get this typical signature of the signal in an experiment on freely diffusing molecules, where every one of these bursts here corresponds to a molecule diffusing through the focus. And the typical duration of these bursts is on the order of a few milliseconds. And now, if you measure inside these droplets, then you also get these bursts of photons. But you can already see, this is on the same time scale, that here the bursts are much longer because the molecules diffuse much more slowly through the confocal volume. So that's already an obvious signature of the high viscosity inside the droplets. And so the key step was to be able to do these nanosecond FCS experiments inside the droplets. And you can see that the signal here is relatively noisy, but it's good enough to get this time scale of the correlation, and it's on the order of 380 nanoseconds. So if you remember the time scale of the one-to-one complex that I showed you of about 100 nanoseconds, it's only a factor of 3 to 4 slower in these droplets, although the bulk viscosity is 300 times greater. And so the question was, why is that? Why do we have these rapid dynamics inside this viscous droplet? And one aspect that those of you have heard about, were there on Friday, is something that we figured out using large-scale MD simulations on the Swiss supercomputer. And so for this, I would like to point out that inside these condensates, we have a very high concentration of charged groups. So if we just count the number of charged side chains in the droplets per volume, then we end up with a concentration of about 1 molar of charged side chains. And so what happens here is that in many situations you have something like this. You have a salt bridge here formed between a basic residue and an acidic residue. And then here comes another basic residue, and they form sort of a ternary interaction, and the original side chain that was making the contact here gets kicked out. And so they constantly replace each other. And the result of that is that we have timescales of these salt bridges that are only on the order of a few nanoseconds. So this rapid exchange between the interactions seems to contribute to the rapid dynamics in these systems, even though we have this large bulk viscosity. However, there is another aspect to it that I didn't mention on Friday, and I'll take the opportunity to mention it here, which is that viscosity in such a complex network fluid is no longer a length scale independent quantity. Right? So in a simple fluid, we can approximate viscosity as sort of a constant thing, independent of which length scale we look at. But if we now do experiments as a function of the size of our probe particles, then we see that there is a highly non-trivial behavior that's going on here. So the viscosity that I was talking about before is what I labeled here as the macroscopic viscosity. So up here, that's our 300 millipascal seconds. And so if you use relatively large beads and you measure their diffusion in the droplets, and then you take those diffusion coefficients, and then using the Stokes-Einstein relation, you can convert that into so if you know the size of your particles and you measure the mean squared displacement, you can infer the diffusion coefficient, right? And so if the beads are large enough, then what you get is the bulk viscosity. But if you use smaller beads or molecules like dextran, for instance, Or in this case, what we did is we measured the rotational motion of a single fluorophore inside the dense phase, that you can see you're actually starting to approach the solvent viscosity, right? So depending on the length scale you probe, you get very different apparent viscosities. And so the key quantity that sort of tells you whether you are locally probing or globally probing the system is the correlation length indicated here. And we can estimate this correlation length, which is something like, in our system, something like the average distance between the polymer chains in our dense phase. We can estimate this based on the size of our molecules inside the dense phase, which we can measure from our single molecule FRED experiments, and the concentration of protein relative to the overlap concentration. And it turns out in our system we're actually pretty close to the overlap concentration. And so we can estimate this correlation length, which is here in the range of a few nanometers. And now we can try to describe this quantitatively, and this is something that I was very surprised about. When we just use these three parameters, you use the solvent viscosity, you use the bulk viscosity, and you use this correlation length and plug it into this theory developed by Rem Couturier almost 20 years ago based on depletion interactions, you get this dashed curve. So this is not a fit. This is just a calculation based on these three parameters. And it does remarkably well in describing this transition from the short length scales to the long length scale dynamics that you have in this system. And if we now look at where is our prothymosine alpha, which is the molecule that we're probing experimentally, it's right here in the range of the correlation length. It's actually, it turns out to diffuse slightly more slowly than these dextran molecules, for instance, of similar size, but that makes sense because it's part of the network, right? It's interacting with the positively charged guys in the system, and so it's diffusing more slowly. But clearly on the length scale of our protein, the dynamics are accelerated because locally the chain can move without having to move through the whole network formed between these different molecules. And so it's the combination of this length scale dependent viscosity on the one hand and these rapid dynamics from the exchange between the side chains that enable these fast dynamics of the IDPs inside their dense phase. Now this is what we can measure with these nanosecond FCS experiments, but there are some limitations of these experiments. And so two of the key limitations are, on the one hand, measuring dynamics that are faster than about 10 nanoseconds is very difficult, because if you remember this correlation function, we had this dip at very short time scales. It's in the range of a few nanoseconds. So if you get too close to that time scale, we can no longer measure the dynamics. The second aspect that I haven't mentioned, from an experimental side, is pretty tedious because these measurements take on the order of 10 hours to get enough photon statistics. The reason is that the average photon rate that we observe in these experiments is relatively low. And so the probability of having photons spaced by nanoseconds is just intrinsically pretty low. And so we need to collect data for a long time to have enough photon data that allow us to get these correlations. And so the question is, can we do better than this? And this is something where Mark Nürsch, PhD student in the lab, decided to use nanophotonic enhancement to improve this situation. And so the idea here is to use zero-mode waveguides. So zero-mode waveguides are nanostructures that are formed simply by, essentially you take a glass cover slide and then you deposit a 100 nanometer layer of metal on top of the glass cover slide, and then you make little holes into the metal layer, and you end up with these 100 nanometer holes in this metal layer on the glass cover slide. And now 100 nanometers is much smaller than the wavelength of light, which means if you now shine light onto the structure from the bottom, it will not be able to penetrate through the system because there are no propagating modes. It's like, I don't know, like the grid in your microwave, right? The microwaves cannot come out because the holes in the grid in your microwave are smaller than the wavelength of the microwaves. And so as a result of this, what we have is that especially here at these edges, we form a very high local electric fields. And so these very high local electric fields enhance the fluorescence from our molecules. That's one effect. So this gives us much higher photon rates. So we can measure much more quickly. We go from measuring for 10 hours to measuring for 10 to 20 minutes. And the second interesting effect is that because of the Purcell effect, we have energy transfer into the metal to some extent, which reduces the fluorescence lifetimes. And if we now compare measurements of similar duration without the zero-mode waveguides and with the zero-mode waveguides, you can already see the qualitative difference in terms of signal to noise, right? I mean, here, this would be a measurement for about an hour without zero-mode waveguides, and would be very difficult to get quantitative data from this. But with the zero-mode waveguides, we can very nicely see the three different correlations. And secondly, if we now look at this photon anti-bunching, right, this peak here at short times, you can see, especially here for the red signal, the acceptor correlation, here it's relatively broad. And because of this reduction in fluorescence lifetimes, this peak becomes very sharp inside the zero-mode waveguide, so we can resolve much faster dynamics. And so in this case, we end up with dynamics just below 10 nanoseconds, and the fastest things that we've measured in this way are down to about 4 nanosecond dynamics. So this now allows us to do experiments on systems that previously we were not able to do. For instance, on short IDPs, like this disordered peptide here only has about 20 residues, and so the dynamics are so quick that with our classical approach we were not able to measure it, but here with this approach we now can. And another opportunity, of course, is if you have such fast dynamics, it's almost simple to compare now to atomistic simulations, right? And so here this is a simulation done by Miloš Ivanović in collaboration with Robert Best, where he simulated this peptide with the fluorophores attached, and he simulated this for 16 microseconds. And so this is 3 orders of magnitude longer than the reconfiguration time, the chain dynamics of the system. So you really have very extensive sampling, and you get beautiful distance distributions with all of the different types of structures that you can get in this highly disordered system. And you can also, for instance, what we have to take into account is that the 2 fluorophores can actually touch each other. If they touch each other, they no longer emit any photons, and we can eliminate this population from the simulations, and then the simulations are in very good agreement with the experimentally measured transfer efficiencies. And what we can also do now, since we have these explicit fluorophores, we can in a very simple way also simulate the photon emission, right? So on top of this MD simulation, you essentially do a Monte Carlo simulation to simulate the photon emission, and then you can directly compare to experiment. And so for instance, this is now a comparison of an experimentally measured correlation function with our dynamics here on this time scale, and this would be from the simulation. And you can see not only is the time that we get from the simulation very close to the experiment, but even the amplitudes of these correlation functions are in pretty good agreement with the experiment, which sort of tells us that we are now at a state where MD simulations can really give us quantitative comparison with experiment. And so from this, we can get at the molecular details of the motion on these systems at a level that has not been possible even 10 years ago. Now, these were examples on disordered proteins. Now, recently we became interested in also trying to do such experiments on nucleic acids, whose dynamics in this range are much less explored than disordered proteins. And so, again, Mark Nürsch in the lab, he decided to study these relatively short, that's about 20 bases of single-stranded nucleic acids, both RNA and DNA, and he used these homopolymeric sequences, and the question was, what's the dynamics in these single-stranded nucleic acids? And given the pronounced base stacking, you might expect that the dynamics are relatively slow, but it turns out that if he does these nanosecond FCS measurements, he gets timescales on the order of 10 to 20 nanoseconds, depending on the sequence. So these dynamics are very rapid. But again, we have the question now, since what we're measuring here is fluorescence, we still need to relate this to a distance, right? And so, unfortunately, the quality of force fields for nucleic acids is much worse than for proteins. And so a few years ago, in a collaboration with Gerhard Hummer, they tried several of the state-of-the-art nucleic acid force fields, and it was very surprising what kind of structures came out of these simulations, I must say, right? I mean, for double-stranded nucleic acids, I think we're on relatively safe ground, but for single-stranded nucleic acids, it was a disaster. And so we decided that we have to use a slightly different approach. And so one possibility would be to, as we were able to do for our disordered proteins, just use some of the commonly used polymer models, right? And so here we have a plot similar to what I showed you before. This would be the transfer efficiency histogram that gives us information about the average distance between our fluorophores. Here we again have the fluorescence lifetime. And so in this two-dimensional plot, here we would have the behavior that would correspond to several different polymer models, a Gaussian chain, a worm-like chain, a excluded volume chain. And you can see none of them sits on top of the experimentally measured data, which would correspond to the white points here. Right? So the polymer models don't do a good job. We need to do something else. Brute force MD simulations don't do the job. And so what we decided to do is, in a collaboration with Gerhard Hummer and his student Lisa Pietrek, we used what they recently developed, a hierarchical chain growth approach. So what they do essentially is they simulate very short fragments of 2 or 3 of these bases, and then from those fragments, they assemble larger structures in a hierarchical approach, and then they can generate these ensembles and then also attach some fluorophores to get the right distances. And it turns out if they do that, this is the result they obtain. So it's pretty close to experiment, but not quite there. So to give it the last little kick, we combine it with a little bit of ensemble reweighting, and then we just reweight to within experimental error of our experimental observation. And then if we do that, we can get a distance distribution as before, then convert this distance distribution again to a potential of mean force, and using this approach from Irina Gopic and Attila Szabo, combine the information from the correlation functions and the transfer efficiencies into a distance correlation time, this tau r, which in this case it's very similar to the fluorescence correlation time because the transfer efficiency here is very close to 0.5, and in this range, distance and transfer efficiency are nearly linearly related, so the difference between the two times is not very large. But if you have systems that are very compact or very expanded, then these corrections are much larger. But the bottom line here is these nucleic acids have a very fast dynamics, and this would have been difficult to measure without the zero-mode waveguides. And so the question is, can we learn more about the nature of the dynamics in the system? And so an approach that we've been using in the past also to investigate the dynamics of IDPs is to look at what we call internal friction. So in these IDPs, we typically see two components of friction. One would be the friction with the solvent. So if we increase the viscosity of the solvent, then the dynamics will slow down, and these are examples of measurements of our correlation functions at different viscosities, and you can see the dynamics slow down. And then what we can do is we can plot these dynamics, these chain reconfiguration times, as a function of solvent viscosity. And for IDPs, what we often observe is that if you extrapolate this to 0 solvent viscosity, then there is an intercept here. And this intercept corresponds to what we call the internal friction time. And if you phrase this in terms of a Rouse model of polymer dynamics, then you can imagine you have two components, right? In the Rouse model, you have beads connected by springs, essentially, right? And so there is friction on the beads moving through the solvent on the one hand, and this would be this time scale here, which just depends linearly on viscosity. But then you can introduce another component that corresponds to friction between the beads, which in a real protein or a nucleic acid would depend on sort of interactions within the chain that slow down the dynamics. And it turns out you can introduce this rigorously into the Rouse model, which ends up being this frictional time here, which is nothing but the intercept in this kind of depiction. And it turns out that for the nucleic acids, for all of the sequences that Mark looked at, they always extrapolate to within experimental error to 0 internal friction. And this was a surprise for me because I would have expected that interactions between these bases would lead to a slowdown of the dynamics. But apparently that doesn't seem to be the case. And so if we look at these ensembles generated in Gerhard's and Lisa's chain growth approach, then what we often see is that these structures are made up of segments where the bases are stacked, but they are separated by hinges where there is no base stacking. So essentially you have sort of solid things that move relative to each other around hinges, and so they move through the solvent, and maybe that's why they are dominated by solvent friction, and we see very little of this kind of internal friction. All right, and with this I would like to summarize. I showed you how we can use our single molecule methods, and especially this rapid correlation spectroscopy, to measure the dynamics of disordered systems, both disordered and unfolded proteins, but also nucleic acids. And we can do this over a broad range of conditions. I showed you the example of the crowded environment in a dense phase droplet. We can also do this under crowding conditions in vitro using just artificial crowders. We've also used it inside live cells to measure dynamics under cellular conditions, which I didn't have time to show you, but it turns out to be very similar to what we see in vitro, so nothing magic happens in the cell so far. Then I showed you how we can improve this type of technology by using nanophotonic enhancement that reduces our data acquisition times and the time resolution that we can get to probe really fast dynamics that we can compare very well to molecular dynamics simulations. And so this shows that on the one hand that we have reached a stage where, at least for disordered proteins, the force fields are at very good quality now that allow us to do quantitative comparisons very well. It allows us now to look at these systems in much more mechanistic detail by combining experiment and simulation. On the other hand, it has already become clear that the single molecule data are a good benchmark for simulations, and so many of these experiments have become systems to check the quality of force fields. And I think that this is sort of a great development in this field, that we can combine theory, simulation, and experiment to understand these systems in much greater detail. And I would like to end by thanking the fabulous team that contributed to this work. I showed you today especially work by Milos, Nicola, and Mark. Alessandro and Madeleine started the work on H1 and prothymazine now almost 10 years ago. Andrea was involved in some of the kinetics that I showed you in the very beginning. Peter and Davide investigated the interactions between H1 and prothymazine on the nucleosome. Daniel Nettles is a key person in the lab for instrumentation, data analysis, modeling. All of the simulation work is a close collaboration with Robert Best, and the zero-mode waveguides we started together with Jérôme Wenger at the Institut Fresnel in Marseille. I mentioned the collaboration with Gerhard Hummer and Lisa Pietrek in Frankfurt, And I didn't have time to talk about the NMR data with Birgit Krachelund and her team in Copenhagen, but this was an important ingredient to understand the system. Thank you very much.
Much for the very nice presentation. We have time for a few questions.
Thank you. It was a great talk. Thank you. My question is, like, you're talking about all these very charged amino acids or nucleic acids. What is the influence of the media, like the salt? Like, is there much influence on that, like in these dynamics? Because I would expect that, like, magnesium would make the last system more rigid, more friction.
No, this is a very good point, and that's actually something I didn't mention. But one of the key driving forces for these highly charged complexes has a lot to do with salt. And unfortunately here I have to sort of, oh yeah, so here we go. One of the key driving forces for this interaction is the entropy of counter-ion release upon binding, right? So each of them individually have their counter-ion cloud. If they bind to each other, then some of these ions are released, and there's gain in translational entropy of these ions, and this seems to be an important driving force because the affinity is extremely dependent on salt concentration. So just to give you a number, if we go from, say, 150 millimolar salt to 300 millimolar salt, the KD changes by 6 orders of magnitude. Yeah? And the binding between these two molecules is actually endothermic. If you look at the temperature dependence or you do calorimetry, you find it's endothermic. So entropy clearly plays an important role. The difference between monovalent and divalent ions in this case is not very pronounced. The divalent ions just act like they just count in terms of their charge, right? So there doesn't seem to be a very specific type of interaction of the ions, but if you just have a divalent ion, you just release 1 ion instead of 2. So you can just account for that. But ions clearly play a very strong role in this type of interaction.
Excellent talk. So let's give a little background to my question. So about 17 years ago, I started working with Vitor on diffusion and protein folding, inspired by your works on cold shock protein. We had a paper on that.
Oh yes, I remember.
And then we went into the ribosome, because where I worked in the ribosome, we did a lot of studies on diffusion as well. So over that time, I've been telling people, the experimentalists, why are you using these cameras that give you this 10 millisecond time scale? You should talk to Ben Schuler and learn about diffusion. And we want experimentalists to, talk to with these questions as well. I doubt any of them have talked to you about it. So but maybe they have. Have you thought about looking at other systems like the ribosome with these diffusion questions, or is it just impossible?
Yeah, so this is something that we tried very hard for about 10 years. The idea was to really try and co-translationally incorporate these fluorophores so we could look at co-translational protein folding. But it was a sort of a very trivial, in a way, biochemical problem, right? Just getting these large dyes incorporated into the chain is just something that nobody has really gotten to work so far.
But how about the ribosome itself? Not proteins, but like the tRNA molecule or the subunits, because they have dyes on those. That's what we're looking at. I wasn't sure if you thought about it.
Okay, yeah. So, I mean, that in principle should be possible, right? So the nice thing about these measurements is that you can, in principle, do it on very large systems, right? I mean, the reason why many people use cameras is if you use immobilized molecules, then cameras allow you to look at many molecules at the same time. Whereas what we have to do is we have to look at every molecule sequentially. And so if you want to sort of start a reaction and then look at many molecules simultaneously, then these camera-based approaches are just very useful. And most people think that everything that's slower than 100 milliseconds is not biologically meaningful anyway, right? So which of course is nonsense. But I think it's just got a cultural problem to some extent.
Could you please talk to them and get them to do what you're doing?
I'll do my best.
We have a question from some of the people watching online. So Anna Povinelli, she asked, first, wonderful talk. I have 2 quick questions. Is the correlation only related to distance, or can it also be influenced by the orientational factor, the kappa squared, particularly in high viscose samples? And the second question, you mentioned the alternative of the zero mode technique to measure dynamics in the low nanosecond range. Would such systems be more accessible with the development of shorter lifetime dyes?
Yeah, two very good questions. So the first question, how do we know we're measuring distance here? And so for this, actually, the fact that we see the correlated behavior in the autocorrelations and the anti-correlated behavior in the cross-correlations is key. So there are other processes that can give you a signal on this time scale. So for instance, if you had dye quenching, or one of the dyes quenching with tryptophan in the protein, you could also get correlation signal on this time scale, but then the cross-correlation is also correlated. We have seen cases like this can happen, so it's important that we see this signature. Similarly, if we have rotational motion in the system, then this can also show up, and I can actually show you a signature of this here in the measurements in the dense phase, right? So this is a situation where you have some influence of rotational dynamics, and there's this funny spike here in the middle of the correlation function where there should actually just be a drop, but there is a funny spike, and this funny spike comes from rotational motion of the fluorophores. So there seems to be some slowdown of the dyes on this time scale that we can see in the correlation function. Here we're lucky that it's separated from the distance dynamics, but it is there. So this is something we can distinguish by, for instance, looking at data taken with polarizing beam splitters and 50-50 beam splitters, so we can distinguish these two components. So that was the first question. The second question, faster lifetime fluorophores would indeed be very useful. However, it turns out that most of the fluorophores that are suitable for single molecule experiments have essentially the same timescales. So all of them are in the range of 2 to 4 nanoseconds as a natural fluorescence lifetime. And there seem to be some intrinsic properties of these fluorophores that it is somehow not possible to get them with much shorter lifetimes, but still high quantum yields, right? So you have things that have shorter lifetimes, but then they have very low quantum yields, and then they're not useful for single molecule spectroscopy. So unfortunately, that has not been an avenue that we have been able, or anyone has been able to use.
Okay, so thank you very much. Fortunately, we have to stop. Okay. Of course, I cannot say no to the Nelson.
This was an absolute spectacular talk, both scientifically and the tour de force to get these things done. What I'm really curious is you're doing these things with extreme high precision, right? I look things like 380 plus or minus 39. How much should I believe these numbers? If I repeat the experiment, if I change the chromophore, so at this point that you have all this, can you tell me how much?
Yeah, so I think this error bar is not unrealistic. Order of magnitude 10% is sort of, I mean, this error bar that we quote is actually not the error bar of the fit or something, right? The largest uncertainty is in how you analyze the data, how you take into account.
If you change your chromophores a little bit, to that
if you change them, yeah. So we have systems where we've done this with different chromophores, and we get essentially the same time scale. So this is within the range of 10-20%. That's the accuracy, not the precision. So
that's
I think
amazing.
That's a reasonable uncertainty for
Thank
what
you.
we can measure.
Sorry to cut things short, but it's just because they are going to use this space for another thing. So let's first thank you so much for the wonderful talk, and he's going to be around, so I think we have more time to discuss. And then we do have a presentation, one of the selected poster presentation by Ander Pereira, who is a PhD student from Unicamp. And I think we may have to shift the space. Vitor? I just heard something very quickly. Vitor? Can we have it here? I'm sorry, it was very quick. I couldn't follow. Or do we have to go somewhere else? What is the situation?
Yeah, I'm sorry. The thing is that they had scheduled regularly to have this space that they use for some other stuff, and so what they asked us, and what I suggest is that we move the next short talk to right after lunchtime, and then we'll have our coffee break, the next coffee break, shortened by 15 minutes, and we'll catch up. So we go now for lunch. Okay? Thank you so much.